So today we'll be looking at the continuation of our class on industrial microbiology. Can you all hear me clearly? Please, can you all hear me?
Yes, sir.
Yes, sir, we can hear you.
Okay. We want to look at monitoring of microbial growth. Give me a few seconds. Let me adjust my screen, please. Can you see my screen?
Yes, sir.
Yes, sir.
Yes, sir.
Please mute yourself as we continue the class, please. So we talked about. Can we all mute ourselves, please? Let me see. Please mute yourselves. I don't want us to have interference, please. I want to mute my own mobile too. So we were looking at other aspects of assessment of microorganisms during their growth. We looked at the growth characteristics during log phase, lag phase, stationary phase, and dead phase. Now, in a typical industrial setting, when microorganism is being used for production process, there's a need for the monitoring of the microbial growth. Who are those people with background noise, please? So then I said in this process, when the microorganism is being used for industrial purpose, there is a need for such growth to be monitored. Typical use of microorganism occur during fermentation because during fermentation, they produce metabolite. These metabolites are responsible for the various applications. Now, there's a need for the monitoring of this growth in order for you to be sure that your organism is viable and your organism is actively at a growth phase that can lead to production of your desired metabolite based on the input, that is, the ingredient that is present. Now, in this process, microbial growth can be monitored. And monitoring of microbial growth is usually by assessment of their cell number or biomass. This allows us to validate their viability and to monitor the process, the progress of the fermentation process. Now, we have an indirect and direct method of monitoring of microbial growth during these processes. The monitoring of microbial growth depends on several things. If you are going to use a direct method or indirect method, it depends on the organism that is being analyzed. It depends on the growth rate of the organism. It depends on the nature of the organism. Is it a filamentous organism, such as fungi? Because fungi, they also produce metabolites that can be used industrially. So if your organism target organism is fungi, so the method to be adopted will depend on a filamentous or non-filamentous bacteria, organism. There are some bacteria that also grow in this format. We have the Streptomyces. So it depends on the nature of the organism that is being analyzed. It also depends on how quickly the result is needed and the degree of accuracy that is needed for the result. The degree of accuracy in terms of how concentrated the organism content against the metabolite, so that there should be no other organism metabolite that is present. In the case where we use a pure culture, there are times that this pure culture may be with other organisms. Then the nature of the sample being tested. These factors are actually the method to be. These factors are to be considered during the process of microbial growth monitoring. So, like I said, we have a direct method and indirect method of monitoring of microbial growth. So, for the direct method, we have simple method of serial dilutions, filtration methods, dry weight determination, cell counting by microscopy, and plate counting methods. These methods are direct methods which can give us direct assessment of microbial growth. It helps us to know that an organism is actually viable, an organism is actually growing, an organism is, actually at a particular phase of growth. Also, we have indirect methods that uses turbidimetry, spectrophotometry, estimation of cell components such as protein, DNA, RNA, and online monitoring based on production of certain gases such as carbon dioxide or utilization of oxygen during their growth. Now, these indirect methods does not deal with the bacterial cell directly. It deals with the activities. The activity in terms of their colony forming, their biomass, or their cell components, or the utilization of certain components or release of certain components. Like turbidimetry, this method relies on the total turbidity, which represents the way at which the organism is growing rapidly. Also, this method is related to spectrophotometry, which also measure the level of absorbance. I think we talked about this previously. Then also, we have the estimation of cell components, which determines, which explain how these organisms are actually growing. Then online monitoring.
I think you
I
checked
will
on
remove
your
some people from my class. Please, I don't want you to disturb this class. If you are not for this class, it's not compulsory. As I was saying, this method actually doesn't deal directly with the organism. It deals with like secondary aspect of the organism. So now we look at examples of the direct method. We have a popular serial dilution, which everybody knows about. Serial dilution of bacteria. How this looks like. This is a year 2 explanation, but it takes a responsibility for us to mention how this looks like. So counting by serial dilution is a direct method because we are looking at the colony and we are going to count it directly. We also are counting by filtration, which helps us to also count the colony of the organism directly using filtration method. We also have direct microscopic counting, which is by the use of counting chambers, which allow us to count using different type of counting chambers, but this time we can have to put the colony in the chamber with a cover slide. Then we calculate based on the colony that we found on the diluting factor. Now, direct method. In the direct method, we have other counting method. Example, we have hemocytometer. The hemocytometer is a type of direct method that uses microscope. It has a grid edge into a counting chamber, and usually a diluted sample of the microbial culture is counted. There's a diluting factor which is used at the end of the counting, and the specific number of square that is counted gives estimation of the microbial cells. So it helps us to count the cell based on the diluting factor in a specific square. So this hemocytometer is not only used for microorganisms, it's also used for other industrial purpose, but it's applicable for microorganisms. So this hemocytometer uses a simple method of counting. We also have Petrol-Hauser counting chamber. This counting chamber are typically used for bacterial counting, can also be used for sperm counting. But we are talking about microbial counting, so we restrict the application to microbiology. It is used in a way of counting grid as well, and it uses a cover slip with a known volume culture, just like where we have the hemocytometer. And the counting is within the proportion of the grid. I don't know those who are disturbing my class. Please mute yourself. Now, by counting the number of cells within the proportion of the grid, it is easy to determine the estimation of the bacterial cell, because you can't count a whole bacterial cell. These are in millions. But when we dilute it, and we know the diluting factor, and we have a fraction of a grid that is counted, the grid can allow us to count the organism. We can count like 80. If you are able to count 80 colonies in the grid, then we'll be able to now estimate based on how we dilute it. And a typical Petrov-Hauser counting chambers look like this. This is how it looks like. A typical one, you can see if you are going to count, we have for the first grid on the left, 1, 2, 3, 4, 5, 6, 7, 8, 9. If you look at this 9, we can easily look at the formula of colony forming unit, which is usually the number of colonies multiplied by the diluting factor over the number of plates. Then we can give an estimation of the bacterial cell. This is how the counting chamber looks like, and it allows us to count based on grid, those that appear within the grid. Like I said, we can use this formula, which is the colony forming unit per mil, against the diluting factor. Example, if you have the bacteria, which is bacteria per mil, such as, let's say, 130 times 10 to the power 6, which is actually the diluting factor. And that will give us this value. So we can say at a particular grid, we have 130 million bacterial cells. It is not possible for us to count this, but we can give an estimate. It is not possible for estimation grants us the privilege to have an idea of the population of the bacterial cells. In each cases, if we are looking at having a lot of bacterial cells, so we look at, oh, we have a 130 million bacterial cells growing at this particular hour. So we can estimate at maybe after 4 hours, we will have had, with multiplication at their geometric rate, it can give us an insight on the total number of colony forming units and what we expect to get. So it can give an insight into the product that will be yielding for an industrialist at a particular period of fermentation process. So this is the reason why this monitoring of microbial growth is essential during fermentation. Because if we discover that the colony forming unit is quite low, then we have to look at what are the reasons for this decrease in the growth in order for us to look at how to provide solution. We have electronic counting method, which also gives us a rapid information on the microbial cells. It gives us a rapid result, and it usually minimizes human error which are associated with other method of counting under the microscope, because counting under the microscope is prone to error. These are limitations of manual method of counting. They have limitations. However, electronic counting method is more rapid and it minimizes human error. It is more consistent and the results are reliable, even if the results are compared with different laboratories and different users. The results are usually consistent, given the same sample for analysis. And electronic counting method can differentiate between organisms of different sizes. It can let us know if we have a pure culture. If our culture is expected to be a particular species of bacteria in a strain, electronic counting method can help us to differentiate different sizes of microbial growth if we are not looking at a mixed colony. So it helps us to get an insight. And it also differentiates between viable cells and non-viable cells in each sample, which most manual counting chamber may not be able to differentiate under normal circumstances.
That is messy. It's messy here.
Okay. I remove people when they disturb this lecture, please. What are the limitations of electronic counting methods? One of the limitations is the fact that the methods are suitable for larger microbes, not for organisms that are smaller. For larger microbes such as yeast, protozoan, and some algae. So we use them for unicellular organisms because they have bigger cells, unlike microorganisms like bacteria and so on that has smaller cells. So it is not useful, really. It is less useful for estimation of bacterial cells. And one of the limitations is the fact that it also requires specialized equipment and is very expensive. It needs a lot of investment to do this, even though it is more accurate, but it's not found everywhere. And it requires a level of installation of certain equipment that can make it work. And also, the calibration is essential. It needs calibration with particle of known size. That is, because it can differentiate different sizes of microorganisms, that means it has to be calibrated with particles of different sizes. Once it recognizes an organism with that size, then it can detect it. So there are multiple, there are different types of particle size, that are actually allowed. Please, I'm begging people to stop disturbing this class. Okay, let me just, perhaps I might have to lock the class so that nobody gets in again. I don't like the way people are disturbing this class. So these are limitations of electronic counting method. We also have another counting method called culture counting method. Culture is a typical method of counting that uses electric impedance. It uses this principle of the displacement of electricity by microbial cells. When there is a change in the electricity of substance that pass through the aperture of the device, it measures it. It measures it and gives a signal of the size of the cell that gave this change through the solution. What am I saying? That this method usually uses what is called electric impedance as a non-conductive particle, such as microbial cells. Once it passes through the small aperture of the device in an electrolyte solution, it changes the level of electric that is being, transmitted. So the magnitude of the impedance is usually directly proportional to the size and shape of the particles. And this is usually converted to a signal in form of a countable pulse that is being recorded. In other words, when a cell passes through the aperture, it will displace its own volume of electrolyte. When the cell passes through the aperture, it displays its own volume of electrolyte and changing the electrical resistance. So these changes are usually detected and converted to a countable pulse and recorded as viability and growth of the microbial cell that is being analyzed. The limitation of this is that it may not be sensitive enough for very small microorganisms such as bacteria again. So when we have microbe cells as small as bacteria, it may not be sensitive for it. It may be sensitive for yeast and some unicellular organisms, some algae, and so on, but may be less useful for bacterial cell because of the level of sensitivity that is based on the sizes. This is a diagram, a schematic function of what I'm talking about. You look at it from here, we have how it works from the valve we have the aperture where the microorganism is passing through. Let me use laser. Now the aperture, we have the valve here where we can get the cell in, and from here we have the positive and the negative terminals, which can allow these electrolytes. These are the electrolytes. It allows them to conduct electricity, and this is the aperture here. So once the microbial culture is run through the valve, when it gets to this point, they begin to do a kind of measurement based on the aperture here. Then the signal is sensed and sent through this plate by giving it a pulse. The pulse are now measured. It gives out the pulse, is measured in form of a signal. And there is an analyzer. We have a counting circuit here. This pulse analyzer helps to now determine what is there. That is just a schematic show of the culture counter. We have the flow cytometry counting method too. This method uses the principle of fluorescence dye. Uses laser and fluorescence dye to identify and count individual microbial cells, which is based on their size, the level of light dispersion, how they can disperse light, which makes them to, kind of fluoresce. And they have a specific signal, which is the fluorescence signal. So this flow cytometry can also be used to differentiate between live cell or viable cell and non-viable cell, in the way that a viable cell will typically give out a fluorescent signal, while a non-viable cell will not give any signal. So the total population of fluorescent signal indicates the total number of viable cells. So in the case where we have a constant rate of microbial death, in this process, it is easy for us to detect that the microorganism is actually entering into a death phase. Maybe there's a limiting factor, maybe there's a decrease in the availability of nutrients or something. Then this allows us to quickly survey the situation so that the fermentation can carry on successfully. This is also the schematic of the flow cytometry. It has a nozzle, and the sample is directly passed through this funnel. And we also have the sheet fluid, which allows the microorganism to mix. And a laser beam is set at a particular specific angle when the sample is passing through directly in this format. This laser light touches the microbial cell. If it's a viable cell, it will give a fluorescent light. If it's not, it will not give a fluorescent signal, because only a viable cell will be able to provide a fluorescent signal. non-viable cell may not provide a fluorescence signal. So this counting method allows monitoring of microbial growth. So it is not just enough for a microbial growth to be counted. It has to be monitored if they are actively growing. Their non-growth is indicated by decrease in the population of active cells and increase in the population of non-viable cells. And when we have more of a non-viable cell, there's a possibility that our desired process, such as maybe antibiotics production, may not be possible after the investment of various supplements to allow for the microbial cells to grow and excrete some of these useful metabolites, which may lead to loss of the. Who are those disturbing my room? Excuse me, please. I think we are matured enough to know that we should mute ourselves at this point. So that is all for the microbial growth monitoring. Then we look at counting method, plate counting method. We will not stay long on this plate counting method because this plate counting method have been discussed in our last class during quality control. And if I remember, most of you took part in the class. So these plate counting methods are used in detecting viable cells only. This plate counting method, they are used in detecting viable cells, those that form colony on appropriate nutrient medium. So we have two main methods, which are pour plates and spread plates. Both methods have specific application based on the nature of the organism again and the type of growth pattern of the organism. For pour plate method, this method is good for bacterial cells. It's good for bacterial cells, and it is not usually suitable for some bacteria that are obligate aerobes, which is suitable for most bacterial cells. While spread plate method is suitable for aerobic and fungal cells. Pour plate is not suitable for fungal cells because of the way and pattern which they grow. Because, like I said, the pour plate method is good also for anaerobic bacterial cells. It allows them to grow easily, but it may not be suitable for obligate aerobes. For the spread plate method, the usual process after serial dilution of the target bacterial cells, it is usually spread on a suitable medium. The final diluting factor, the final diluting concentration, is usually plated on a suitable agar medium. And a glass spreader, sterile glass spreader, is used to spread, that's what they call it, spreading. Used to spread it on the plate. And this plate is now incubated. usually incubated by inverting the plate, that is turning the plate upside down and allow it to grow at optimum temperature for the target microbial cell. With fungi, temperature has to be lowered to around 25 degrees. With microorganism, it has to, if it is bacterial cell, it has to be higher. I wrote there, take note that microorganisms with low tolerance to oxygen may not grow well unless it is incubated under appropriate conditions. So appropriate condition has to be applied for this. For pour plate method, the pour plate method simply follow the same procedure of dilution and taking the last diluting concentration. But in this method, the volume, like 1 ml, just like we have the previous method, is now either introduced into a molten agar and allowed to distribute evenly. It can be put in let me say 1 ml of the diluted concentration can be added to a bottle of McCartney containing 20 mils and swirl constantly for even distribution. Then it is now poured. What I mean is that we have some bottles, call them super bottles or McCartney bottles, that can take up to 20 mils of the agar medium, which is still molten. So when this is allowed to cool to about 40 to 30, let me say 40 degrees, which is not yet solidifying, microbial cell is now introduced. 1 ml of it is introduced into it, and it is gently mixed together, then poured in a petri dish and allowed to solidify. Some method can actually pour the last 1 ml in a plate and pour the molten agar on it and gently swerve the plate also for evenly distribution. Then it will now solidify. After incubation, the growths are enumerated because it will have a different microbial growth which can allow them to be seen throughout the surface of the agar. So both methods now will allow you to count the organism that grow. Now, depending on how diluted the concentration that is used, you can actually count the organism. You can get to about 30 organisms growing, can get about 5, can get about 50. By using the same formula previously, which is colony forming formula, we can get an estimated number of viable bacterial cells that is present. So for turbidimetry method, this method also we have discussed about this in our previous class, which uses the light absorbing power of the spectrophotometry. The turbidimetry method and spectrophotometry method are a bit similar. They are both using the estimation of biomass in each microorganism, whereby it allows to measure the rate of light that passes through the cell. The spectrophotometry allows us to measure the total absorbance of light based on the standard that is that is available. And once we can measure the absorbance, we can determine how turbid the microbial cells. The more turbid the microbial cells, then the more you can say the organism is growing. If the organism concentration is less turbid, then we can say the growth is lesser. We have another method, which is online estimation. Online estimation for fermentation of microbial cells provide real-time estimation of biomass, and it helps to minimize the requirement for repeated sampling and offline analysis. What do we mean? Repeated sampling is talking about taking a sample at every interval, doing the same method of microbial growth assessment. It minimizes this. It can give an accurate growth per time. It measures it as it's occurring. We don't need to do hourly, 2-hour, 3-hour measurement of microbial growth. It can give us real-time estimation at a particular time. We may decide to check at a particular time at 4 hours. I want to know the total growth. 1 hour, 3 hours. It can give you the estimated growth per time, not by usual previous method of counting. It may involve optical density or capacitor-based probes. It may involve, it depends on the method of the online estimation. Some can use optical density method, which is usually spectrophotometry-based online estimation, or capacitor-based probe, which uses some part of electrical method and introduction of probes that give a signal, like a biosensor. And also, the microorganism in the most fermentation will either have a requirement for oxygen, or they will have to be producing carbon dioxide, which is going to be measured. That is, the requirement for the oxygen, how the demand for oxygen can be measured. When the demand for oxygen at a higher level, then that means their growth is more. When they also produce carbon dioxide at higher level, that means their growth is increasing. So these are principles that guide different online monitoring of microbial growth in online estimation. So these processes uses certain method or principle that is like a form of standard for its procedure, for its working method. And online estimation depends on certain factors. Number one, it depends on desired information, whether the total cell number, size, metabolic activity, or specific type of cell to be monitored. So the desired information will determine what method to be used. If you want to know the total cell number, it's a different method. You want to know the cell size, metabolic activity. Metabolic activity we have to do with some level of oxygen demand and carbon dioxide release, or also specific type of cells to be monitored. So the desired type of information is a factor that has to be considered. Then also the culture characteristics. Culture characteristics is talking about the specific organism and the growth medium. And also, it depends on the cost and complexity of the method required and the accuracy of the data. So it can be costly. And if it's a complex process, then you have to think about the most suitable one to reduce costs. So these are factors that are considered during online estimation method. So we have types of online estimation method. We have types of online estimation method. We have the electrical impedance spectrophotometry. We also have biosensors. We have fluorescence techniques, respiratory measurement. Let me explain them one by one. The electrical impedance spectrometry, also known as EIS, measures the electrical properties of the culture, and this includes capacitance and resistance. Now, in measuring the capacitance and resistance, is there any change in these properties? It is being correlated with the microbial growth, and it also correlates with the cell morphology. Although it is a very complex technique, but it's more complex than optical density and every other method that I use. However, it has its own, it offers more analytics because it measures the change in the capacitance and the resistance based on the microbial growth and their cell morphology. Their cell morphology may determine how they do the resistance change and the capacitance during the electrical impedance. It means that it impedes electricity flow. So once this is done, it can give us an idea of the, it's an estimation. Everything is an estimation. So it gives us an estimation of these microbial cells. Then we have the biosensors. The biosensors, it has a device. It's like a device. These are device. The device actually are made. They are constructed, and they are constructed, but they use biological matter, like an enzymes, antibodies. They use biological matters or elements such as enzymes, antibodies, which can interact specifically with microbial component in the device. So these enzymes can actually interact. Once they interact with the microbial cell, they give a particular transmission of electrical signal. They give signal. The intensity of the signal.
The man was saying that
Oh my God. Oh my God. So they give a particular type of signal which is based on the interaction of these biological elements. And so this interaction can now be transduced into electrical signal, which can provide a measurement of the specific microbial growth per time. Remember, it's an estimate. So as it gives this signal, as it gives electrical signals, it is a way of giving an online estimation of the microbial growth. Although this method is also an estimation method, but it offers a high specificity because these biological elements I'm talking about has ability to specifically interact with the microbial component. When we talk about microbial component, it could mean some part of microbial cells which are kind of indicator of their growth. So once these organisms are growing, these biological matters interact and it gives electrical signals. So we call them biosensors. They are specific in their working principle. However, they require complex development, and also some high level of maintenance because after use of every biosensor, there must be a good maintenance. Otherwise, it will not function properly. We also have fluorescence techniques, part of what I mentioned the other time, but in a way differently. It can use some probes, or this method uses certain probes or dyes, which can be used to stain microbial cells and allow them to fluoresce. They can actually glow and the level of their fluorescence can be measured. It can be measured. This method is also a way of flow cytometry. It's a kind of example of flow cytometry. The same principle of flow cytometry that I described previously is also an example of fluorescent techniques that is for online estimation. Now, the difference is that it may not be applied in the same way, but it can have a specific device that is connected for this purpose. It is also very sensitive and also require specific protocol, maintenance, specialized equipment, personnel, and all that. So this method, as good as they require specialized personnel and high level of training for it to be deployed for use. Then we have the respiration measurement. Respiration measurement monitor the rate of oxygen consumption or carbon dioxide production. Like I told you the other time, this method is a way of determining if an organism consumes oxygen or produce carbon dioxide. So respiratory measurement is also an online estimation type, which allows us to know the increasing metabolic activity during growth of the organism. So the increasing metabolic activity will determine their level of oxygen consumption. If it keeps increasing, that indicates an increase in metabolic activity. If it's decreasing, then that means the cells are dying. So if there is an increase in metabolic activity, it indicates an higher respiratory rate. That means the rate at which the organism are carrying out respiration is increasing. So increasing metabolic activity during growth rate usually leads to higher respiration rates. And this is a form of measurement, because respiration measurement provides a direct measurement of microbial activity. So if we can measure the respiratory activity, it can give us a clue of the microbial cells growth. So these are types of online estimation types. I hope you will not forget these methods. From there, we are going to look at effect of different environmental conditions on microbial growth. Effect of different environmental conditions on microbial growth. We all know that growth and development of microorganisms greatly affect what we do in the industry, and they can also be a kind of indicator of how these organisms are faring during the fermentation process, and their growth may be affected directly, indirectly, by environmental factors such as chemical and physical conditions and their other associated environmental factors. So microorganisms in the environment have ranges of tolerance to different environmental conditions. And these different tolerance are exploited in the industry. Some microorganisms are from extreme environment, which can also be useful. When we say extreme environment, some of them are found growing in high temperature environments. So their cells have been adapted, and they produce certain enzymes that can be used for high temperature processes. For example, we have the PCR that uses the enzymes from certain organisms that grows in high temperature environment. Who can tell us an example of this type of organism and the enzymes? Who want to tell us? You can unmute yourself and tell us. I know you know it, but I just want to know if you remember. Example of a bacteria that grows, that can endure high temperature of up to more than 80 degrees. The enzyme is useful for polymerase chain reaction. Who want to tell us? This lecture is streamed, so don't disappoint me.
Thermus aquaticus, sir. And it
Correct.
produces stack polymerase.
Correct. Good. So that's an example that is being exploited in the industry based on the fact that the organism grows in an extreme environment. So the effect of environmental conditions, we have temperature, we have the pH, we have the solute and water activity, we have the oxygen, we have the radiation, and we have the hydrostatic pressure on growth. These are different environmental conditions that have direct effect on microbial growth. So the temperature is essential for their growth. Every microorganism has its own optimum growth temperature at which all metabolic activity goes unhindered. The optimal temperature of every organism is the perfect temperature for its growth. They grow faster at increasing temperature because it allows the rate of chemical reaction to increase. And this doubles at every 10-degree rise. However, if there's any reverse of temperature, this usually reduces their growth. So, temperature is highly essential for microorganisms. If the temperature is reduced, it may reduce their growth. If it increases their growth, but it must be at optimum temperature. However, there are limits to which microbial cell can take increase in temperature because the cell contain certain temperature sensitive macromolecules such as lipids, even the other cell membrane particles which can be denatured. We also have some of the proteins, nucleic acid. They can actually be denatured and become non-functional if the temperature have gone beyond the limit of the microbial temperature tolerance level. So each organism has its optimum temperature. Lower temperature too will have its own negative impact because if the optimal temperature is not attained, if temperature drops lower, they will not grow because they have the lipid membrane which will not function properly and it will not allow their cells to function properly. Lipid will form solid at lower temperature. That is why some microorganisms can actually be preserved in the refrigerator to slow their growth because once the temperature is lowered, the lipid content of their cell becomes non-functional. Based on the fact that microorganism has only one cell, if there's anything that can alter any part of their cells, it will affect their total growth. Unlike multicellular organisms that can still manage themselves with other cell components, bacteria may not. On this basis, we have different types of microorganism and temperature requirement. We have the saprophytes, which are usually growing at lower temperature. They have the optimum temperature around 15 degrees, and they can often be destroyed when we expose them to room temperature. So the saprophytes are the lowest temperature tolerating organism. We also have the thermophiles. Their optimal temperature is from 50 and they can get up to 80 degrees. We have the stenothermals, which grow over a temperature range of approximately 30 degrees. We have the eutherma, which grow over wider range. We also have several algae, protozoa, and fungal species, which have temperatures around 55 degrees centigrade. And we have some prokaryotes, which are actually thermophilic. They grow at higher temperature. Although eukaryotic grow only at moderate temperature, most of them do not grow as high as prokaryotic. Okay, in the aspect of thermophiles, in the aspect of the temperature, I omitted something. That is, the thermophiles, that those that love to grow at high temperature, they have certain adaptations in their cells, and this allows them to be able to endure high temperature. High temperature usually can destroy a non-thermophile because it will cause damage to their cytoplasmic membrane. When an organism has an optimum temperature growth for 45 degrees, such as mesophiles, when exposed to higher temperature of about 80 degrees, may die. But other organisms that have a kind of adaptation, in the sense that they have their cells, they don't have lipid content that can easily be destroyed in their cells. So their cells have actually been modified in a way of adaptation to high temperature, because lipid content affects bacteria in the sense that lower temperature make it non-functional. Higher temperature also make it not functional. So non-thermophiles usually die. They have their membrane breakdown because of damage to their DNA. Once their DNA is denatured, their strands begin to break and they die. So it is difficult to determine which of their heat injuries is occurring during this high temperature. But actual organisms with adaptation to high temperature, thermophiles, they are able to survive because they have a modified nature, and some of them can actually grow above 100 degrees, and they all have their industrial uses, especially in the production of certain products. So the effect of pH also, the way we have microorganisms that has tolerance for PM temperature, we also have microorganisms that have different tolerance for pH. So we have the acidophiles and we also have the alkalophiles. The acidophiles are those that love lower pH between 1 and 5.5, whereas the alkalophiles prefer pH between 8.5 and 11.5 above. Though we have some fungi that grow at pH between 4 and 6 than most bacteria. However, in terms of loving acid, in terms of pH tolerance, these organisms prefer lower pH, that is the acidophiles, whereas the alkalophiles prefer the higher pH. So, knowing their nature will allow us to determine the content of the medium. Because we know the effect of the pH, so pH is regulated whenever these organisms are present in our fermentation process. If you are working with acid-loving organisms, then it means that the pH of the environment must be lowered for the organism to grow. If the pH is higher, that means the organism may be affected. So in monitoring the growth, microbial growth, after checking every nutrient that is available, the pH is also measured. If the pH is accurate and the microorganism is no longer growing, then it is not from the industrial setup. It may be the fact that the organism is not good or is not an industrial strain. So we also have the effect of water and solutes on microbial growth. The effect of water solute is called water activity. Different microorganisms require different water for growth. We call it water activity. So most microorganisms contain approximately 70 to 80% water, and they require some amount of free water for the specific metabolic activities. So they require these specific metabolic activities for their growth process. In an environment where there is low water availability, the microorganism, even though may contain water, it needs water in the environment for its activity to be optimal. So we have what is called hypertonic environment, which takes which allows the change in the water concentration between the environment and the microbial cells. In this process, water uptake and retention is difficult because the environment, the differences is different. So also is hypotonic. The hypotonic may contain considerable amount of water, but it is not going to be available for the organism. So this process has to also be monitored so that the organism may not lose their water activity. Once they lose their water activity, there is no metabolic process. When there is no metabolic process, organism may not be able to produce desired metabolite for industrial processes. So these factors are very important for microbial growth fermentation process. They are factors to be considered. So these are a process that shows the formula for calculating water activity. Water activity in microorganism is indicated by the term of AW, and it's the ratio of water vapor pressure in the solution surrounding the organism to the water vapor of the pure water, which is calculated in this form. AW, which is water activity, and the water vapor and the pure water. So as of pure water, the water activity is 1. So most bacteria cannot grow below water activity that is below 1. Most bacteria, with few exceptions, they cannot grow below 1. So a pure water activity is equals to 1. If the organism is fungi, especially those that cause biodeterioration, they can actually grow below 1, even up to 0.7. If you know those that cause biodeterioration of stored grain are so dry. They're usually dry. It is only fungi that causes mycotoxin and so on that grows and spoil these substances. You hardly see bacteria there. The only time bacteria contaminate these grains is when there is dampy environment, when the grains have some level of increase in water concentration. At this point, the water is increased and the bacteria can then grow. But fungi, they are usually growing at this temperature, at this water level, at water activity level, and allow them to spoil some of these stored grain. So we have different water level activity for different fungi. We have the Zygosaccharomyces roxy that can inhabit the environment as low as 0.6%. This is very low. Some of them may not grow under condition of higher water availability because they don't require lots of water for their growth. They require just little water, but bacteria require more water. So bacteria usually grow above that water activity. That is why without water in an environment, bacteria may not be able to contaminate and grow, but fungi, they will grow. That's why you see fungi growing on walls. You see the molds, you see them growing on the walls. They damage some of these dry food substances because they require just a low water activity. And these are also measurement of preventing fungal growth because they do not require much water. They cannot tolerate higher concentration of water, but bacteria can do that. We also have the halophiles. In terms of the effect of solute and water, we have the halophiles, and also we have those that are found in salt environment. Salty environment are those that love to grow in high salt concentration. High salt concentration is a way of controlling bacteria. Some organisms may not love this process because the salt content is usually a way of controlling bacteria. But we have some marine bacteria that are optimally adapted to salt concentration in the sea. These kind of marine bacteria, their water activity is usually around 0.8, 0.9, around 0.98, approximately 1. Some of them, they possess enzymes that have a specific requirement for sodium ions so that it allows them to adapt to salt concentration in the seawater. The true halophiles, example we have here, Halobacterium allobium. Those ones are found in areas with salt lakes. They will not grow in salt concentration that is less than 3 mol per liter because of their nature. So we also have the osmophilic, aerophilic, and halophilic organisms. Also, in their nature, they accumulate compensating solutes, which balance the osmotic strength of the external solute in a way of growth. So these different organisms, although most of them, their industrial use are limited. Their industrial use are limited to certain specific products of industrial use or biomedical uses. Rarely do you find them producing some other human use. Some of them produce some antibiotics also. They produce some antimicrobial agents. Not really antibiotics, they produce antimicrobial agents that can be used in controlling some other microorganisms. But usually their use are limited in the industry. So I think, okay. Then we have examples, those that are here. We have the polyols, arabitos, and other filamentous yeast, also that are found in halophilic environment. So many halophytes also store large amount of potassium ions, and some of them, they amass some of these things in their cells. So these are examples of halophytes and their tolerance level. Effect of oxygen. Effect of oxygen is found in types of classification of microorganisms based on their oxygen requirements. This is a known classification. We have the obligate aerobes. Those are organisms which grow in the presence of oxygen, only in the presence of oxygen, as it is required as the terminal electron acceptor for them. They do not grow aside this, they grow only in the presence of oxygen. These are examples of organisms I mentioned that you can't use a kind of counting method for. You have to be specific. We also have facultative anaerobes, which functions whether there is oxygen or without oxygen. Example, we have E. coli. We have E. coli as a very standard organism. We also have Pseudomonas aeruginosa. Some medically important organisms, they are facultative anaerobes. They grow efficiently when oxygen is available. When oxygen is not available, they still grow. So we have other types which are the microaerophilic. Microphiles grow at a very reduced concentration of oxygen, depending on the type of requirement. Some can require 5% oxygen, some can require 10% oxygen. Only require small amount of oxygen for their biosynthesis of certain compounds. So if the organism is increased more than what they can tolerate, they will not grow and they will not have any activity. So they must have a lower oxygen level. Some has about 2%, some have a 10%, 5% oxygen. Example, we have the Campylobacter, we have the Helicobacter. Although Campylobacter and Helicobacter, they are of no industrial use, but we are talking about oxygen, microorganisms that are classified based on their requirement for oxygen. The aerotolerant anaerobes, they are organisms that actually essentially ignore oxygen and they grow well equally in its presence or in absence, while obligate anaerobes cannot tolerate oxygen at all. Obligate anaerobes and obligate aerobes, they are two extreme organisms that may not be manipulated. It's either you give them the oxygen or they don't want the oxygen. So these organisms and their lifestyle are based on the type of requirements. So very quickly, we look at adaptation of microorganisms to oxygen. We have three groups of enzymes that allows organisms to kind of grow freely. It helps them to render their activities. So three groups of enzymes catalyze free radicals to help render them harmless. We have the superoxide, dismutase, catalase, and peroxidase. These enzymes are actually present as a free radicals. Aerobes and other microorganisms, excuse me, please. Someone is asking a question. So what is the difference between aerotolerance anaerobes and facultative anaerobes? Well, for aerotolerance and facultative, you can say they are similar, just some little differences. You see, aerotolerance can actually decide, they can ignore oxygen, they can decide to ignore oxygen and not grow at all. Whereas facultative aerobes. Facultative aerobes may not really ignore oxygen, but if there is no oxygen, they grow. That's the difference. Facultative anaerobes, they function with oxygen or without oxygen, not by choice, like E. coli and stamina. But aerotolerance anaerobes, they can actually deliberately may not require oxygen, even though they can grow well in its presence or absence. I hope I'll be able to explain their question to you. I was somewhere, got distracted now. So I was talking about aerobes and other organisms in their oxygen tolerance. So they have these superoxide and dismutase. Many of them, excuse me. So these enzymes, as I was saying, are present in this organism and it has specific function. For instance, aerobes they possess catalysts for eliminating hydrogen peroxides. Catalysts in this aspect, when you check for catalyst positive organisms, you discover that the reason for this effervescence is the fact that the hydrogen peroxide that is being used as a test displaces the oxygen and hydrogen and gives the gas. So it possesses the ability to eliminate hydrogen peroxide, and this is a way of their own tolerance. We have those that are strict obligate anaerobes. They lack superoxide, dismutase, catalase. So, or they produce them in inadequate quantities. So they have a very limited ability to be able to function in the presence of oxygen. So these enzymes are what determines their activity in terms of oxygen availability or oxygen absence of oxygen. So they may have a very limited ability to detoxify oxygen radicals, and so they are unable to survive in the presence of oxygen because they do not possess the enzymes that helps them in this process. So these are enzymes that allow them to adapt to this environment that is possessive of oxygen and unavailability of oxygen. So the effect of oxygen on growth, as expected, has an impact. So anaerobic habitats are rather more common than expected. They may arise from the utilization of oxygen by facultative anaerobes to create an anaerobic environment. What this means is that in the process of growth, more anaerobic habitats are created because of the utilization of oxygen. The more oxygen that grows, the more organisms that grow and carry on respiration, the more demand for oxygen, the more less availability of oxygen in the environment, and anaerobic environment are created because the demand for it, and some of them are facultative anaerobes. So they create anaerobic environment where obligate anaerobes grow. So in knowing this also, it helps in some process to have some inflow of air for fermentation process. So because anaerobic environment will be created, and if this will be an impedance to growth, so there must be a constant air supply in the fermentation process. So because of the effect of oxygen, it will not allow some of them to grow properly. So in the effect of oxygen, we have the use of media containing reducing agents such as thioglycolates, which chemically bind with oxygen. We also have the physical removal of oxygen. If the oxygen has to be removed from the environment, if oxygen have to be removed from the environment, we can use some reducing agent. We can also physically remove oxygen by pump, also the use of gas packs, and we also have anaerobic cabinet for manipulation of presence of oxygen in the effect of negative oxygen. If oxygen is desired, air can be pumped in. But if oxygen is not desired, these methods are used in removing oxygen from the environment so that the organism can grow and produce what we want. We have the effect of radiation. Radiation usually are rays that can cause excitement in microbial cells, and usually this excitement have a consequence effect that leads to mutation. Eventually, we kill the organism. So in no two ways, we know that this radiation has detrimental effect. So radiation on microbial growth can kill an organism. Especially, we have certain microorganisms, we have Deinococcus radiodurans, which exhibits considerable natural resistance to radiation. So these organisms, aside this, many other organisms are susceptible. They may not absorb radiation. Others can absorb radiation. Others will die in the presence of radiation. Their DNA can be damaged because they do not possess certain adaptive features such as these Deinococcus. So, and if you look at some of these organisms, they don't have any industrial relevance. But the effect of the radiation has a detrimental effect on the organism, and this can actually damage their cells. So microorganisms are less susceptible, while some are resistant. So radiation must be properly regulated in course of fermentation to avoid cellular death. So, and where do we get radiation from? The radiation is usually in sterilizing environment. Some environments which we know popularly in the medical field, the theater usually have a radiation for cleaning the environment. Also in the industrial setting, some of these radiation are also there to remove and destroy bacterial spores, also to cause effect of killing, to sterilize some of the preparatory room. So the radiation must be properly regulated to avoid direct regulation on fermentation, which can lead to death of organism. The hydrostatic pressure. The effect of hydrostatic pressure is a measurement of the depth and height based on how microorganisms can adapt to pressure in depth. When we talk about depth, we're talking about in terms of pressure that is exerted at a particular region. In nature, Microorganisms are never subjected to pressure in excess of 1 atmosphere and higher pressure. The higher pressure generally inhibits microbial growth. So when there is higher pressure, microbial growth are inhibited. And this can affect their protein synthesis and also directly affect their metabolic activity in terms of biosynthetic process. So in terms of production of certain products in the fermenter, the depths are to be considered. If the depth, oh my God, if the depth is too, if the fermenter is too deep, it can exert pressure on the microbial cells. What we're saying is that microorganisms usually they are not, they don't like higher pressure. This higher pressure has direct effect on them. So if the fermenting medium, if the what is it? The fermentation medium and fermentation jar, fermentation, who are these people is just distracting me. If the fermentation um, container has a very deep design and it's run from the depth up, it may affect the activity of this organism. That's what I'm trying to say. To affect the activity because microorganisms do not like higher pressure. So this is also very important. And for example, in cider production, yeast performance is impaired when fermenter deep depths are greater than 14.5 meter, which produce hydrostatic pressure above 1.5 atmosphere. So microorganisms are usually adapted to their optimal temperature should not be pressure should not be above 1 atmosphere. Once we have a depth that can exert a pressure, because if you look at the lower you go down, the more pressure you experience. Even people that dive down, they have what is called some features they use. The scuba divers help them to adapt to pressure. But microorganisms that are in a deep ocean, they already adapted. Their cells are adapted to higher pressure, unlike an industrial stream that is taken from a different environment to come and function in the industry, must not be subjected to such kind of pressure, is the point I'm trying to make here. However, many marine organisms must be barotolerant as they live at depth already. Like I said, those ones are usually subject to pressures of several hundred atmosphere. And so those ones are usually balanced in terms of pressure. But other organisms, such as could be pseudomonas that are found in the terrestrial environment, may not function. Saccharomyces will not function because they cannot adapt this level of pressure. Time is far going, I have to round up on time. So control of microbial growth, how can microbial growth be controlled in the industry? We have the sterilization procedures. These sterilization procedures helps in elimination of microorganisms in the environment. Example is the radiation I have mentioned. These are carried out to eliminate microorganisms in the environment. So we have sterilization procedures that completely destroy or eliminate all viable organisms, including their spores, which are usually tolerant to some harsh conditions. So certain sterilization procedures can be carried out. And we have factors that influence the effectiveness of sterilization procedure by the use of antimicrobial treatment. We have the population size. The population size determines the level of antimicrobial treatment. If the population size is more, then the antimicrobial treatment may not be possible. There are some sterilization procedure that are specific for certain microbial population. For instance, radiation can be used in a room, in a chamber, where fermentation can be carried out. But sterilization of equipment may not be used with radiation. We can use some other selection procedures such as autoclaving, giant autoclaving. Whereas in some, we have to use antimicrobial agents like disinfectant and antiseptic antimicrobial agent that can help to eliminate it. So different microbial population and a specific location will determine the type of sterilization procedure that will be used. But in terms of antimicrobial treatment, population size is critically important. The equivalent of the population of microorganisms as to the antimicrobial agent that will be used must be equivalent. Otherwise, the antimicrobial agent will not be effective. The population composition also varies. Population composition, also determines in terms of a mixed population. There are certain antimicrobial agents that are specific in killing certain types of microorganisms. For fungi, it's different from bacteria, and for algae and protozoa, they're all different. So the population composition is also critically important. Also, is the concentration of the antimicrobial agent or the intensity of the treatment. That is, the minimum inhibiting concentration of the antimicrobial agent must be also factored out, which is the minimum inhibitory concentration that can kill. So if a proper sterilization procedure has to be carried out, that means we have to use concentration that is above the minimum inhibitory concentration. If the concentration that is lesser than the MIC it is used, then we will not have effective sterilization. Also, the period of exposure to the leather agent, period of exposure, the contact time, which is contact time has to be longer. It must not be a short contact time. Contact time is the process at which the time at which the antimicrobial agent is applied to the surface, either surface or container or device, and removed. So the contact time allows more organisms to die. Lesser contact time allows fewer organisms because some are still in the process of multiplying and resisting. So if the contact time is not allowed to extend, it may lead to improper or ineffectiveness of antimicrobial agent. Then temperature is also very important. Temperature also aid microbial elimination. So combination of temperature with antimicrobial agent is very, very important because some antimicrobial agent may be denatured by higher temperature. The use of certain antimicrobial agents in the presence of higher temperature may render them ineffective, whereas the organism may survive. Lastly, environmental conditions such as pH, medium viscosity, and the concentration of organic matter are critically important. Environmental conditions such as pH, like we said, some organisms which prefer to grow at a particular pH must not be treated in that pH concentration. They must be subject to alternate pH concentration so that the antimicrobial agent may have effect. Now, if this is not so, certain antimicrobial agent may not function in the presence of certain pH. So this is also a factor. So for antimicrobial agent, neutral pH is best to be applied, and the treating medium, because sometimes you are not actually sterilizing the medium, you're actually sterilizing certain devices. So the antimicrobial agent must be applied at a neutral pH, especially if it's in a certain fluid that is being expected to be used for fermentation process, maybe the water. Then the medium viscosity also is essential because it may not allow the permeation of the antimicrobial agent. The concentration of organic matter can cause reduction in the activity of antimicrobial agent because the organic matter do not contain the organism, but it reacts with the antimicrobial agent, reducing its killing ability because the organic matter actually reduce the concentration of the antimicrobial agent, making it less available to the microbial cell, which can lead to their death. So more organisms tends to survive in the presence of organic matter, while more organisms die in the absence of organic matter. So these are factors that can influence the effectiveness of any antimicrobial agent during sterilization of industrial equipment, industrial ingredients, everything that is meant for fermentation process. The reason for sterilization is because we don't want undesired microorganisms in the fermentation chamber. Undesired microorganisms will lead to the production of certain products that are not relevant. So if I ask a question, I explain. For instance, if a question is asked you, why do sterilization process a requirement for fermentation? You know what it's going to talk about. And what are the factors? These are things that we talk about. We discuss this and more. There's a way question can be coined in any form. There are different question areas that could be described, but this is just an example. So this is the end of the class. Our next class will be talk about it. But before we go, I have an important announcement